So this is likely something people may want to know and just take into consideration. This was in 2016 from PubMed. Um, Barcoding the brain. Barcoding. Yes, barcoding the brain. I'm not going to read all of this because it's very scientific. It's not very long though, but it's what the title says, Barcoding the Brain. So I've said before that I believe that everybody's already taken the mark. Um, they just want to continue to lead you on this idea that, oh, it's coming, it's coming. They like to play those games. And again, the preachers and pastors taught that certain people won't take it. No, it says in Revelations, go back and read it. This is why we weren't to just follow whatever a preacher said. It says all will take it. Yes, all will take it. Young and old, rich and poor, all will take it. So they wouldn't want you to know it's coming, but they would want to, they love to fearmonger. So they have a bunch of people believing it's coming in this form, in this form. No, it was your mapping your brain. Yeah, that's the barcode. Um, so I'll read a little bit of this. So barcoding the brain, 2016. About 5 years ago, neuroscientist Tony Zader saw a slide at a scientific meeting that changed the course of his career. It showed a slice of a mouse brain tissue containing a bright rainbow of neurons. Under most microscopes, neurons normally look gray and so densely packed as to be indistinguishable. But here, each cell had been genetically engineered to produce random colors. Zadar was struck by an idea. If he could replace the half dozen distinct colors with a label consisting of, say, 30 random nucleotides, the barcodes to an almost infinite number of neurons, then if he also found a way to chemically link barcodes together across synapses, Zadar could use high throughput gene sequencing to build a synapse by synapse map of brain connections, a goal that is dear to neuroscientists who want to understand how the brain's intricate wiring underlies its functions. Unlike existing brain mapping methods, the process could be thorough. Now, after a long slog and a marriage with another technology for tracking the barcodes in intact brain tissue, the researcher at Cold Spring Harbor Laboratory, New York, believes the technique is ready for prime time. A lot of individual steps and problems need to be solved, says Ed Calloway, a neuroscientist at Salk Institute for Biological Studies in San Diego. But the refined version could, in one shot, get all the connections of a single brain. In parentheses, they have mouse. The U.S. government is placing a large bet on Zader's method. Well, I bet they are. As part of a project led by molecular engineer George Church of Harvard University, what a surprise, the technique was selected last month to receive 21 million. 5-year brain mapping grant, one of 6 new projects under the Machine Intelligence from Cortical Networks, or acronym MICRONS, program sponsored by the Intelligence Advanced Research Projects Activity. That's IARPA. Intelligence Advanced Research Projects Activity, IARPA. Part of the U.S. initiative launched at President Barack Obama's request in 2013. Of course. Microns aims to map every neuron and synaptic connection with a 1 cubic millimeter chunk of tissue from the mouse visual cortex. Ultimately, ARPA hopes those detailed brain connection maps will help scientists design computer architectures able to perform tasks that are easy for a brain but out of reach for artificial intelligence, recognizing a person or object from many different angles after seeing it only once, for example. In the early version of the method, Zadar and his team mapped neural connections in mice by injecting viruses encoding nucleotide barcodes into their brains. They would then use a custom-designed chemical to join and extract the paired barcodes on either side of a synapse from the post-mortem brain. Although the technique could reveal which neurons were connected to other neurons, fine spatial information about where cells were located was lost because they had to mash up the brain tissue to extract the barcodes. It was a brain in a blender approach. That's our science. Okay. The companion technique developed by Church, the one from Harvard, the other soulless wonder, solves the issue by allowing Zadar to read the barcodes without the need to extract them from the brain. The technique called physique, F-I-S-S-E-Q, applies washes of different fluorescent molecules to the tissue. Each molecule links to a specific nucleotide, so successive washes cause each letter in a barcode to light up one at a time, like a series of string lights on a Christmas tree. And an optical microscope captures the sequence. If a barcode in a given cell is ATGGCG, for example, the sequence of colors might be red, yellow, green, green, cyan, green. When barcodes from two different cells show up within a synapse's distances of each other, a computer program infers that those two neurons are connected. Zadar and Church predict their joint technique can extract the connectivity of a cubic centimeter of a mouse's brain tissue in days or weeks, not the months or years typical electron microscopy. But hurdles remain. Zadar says injecting the viruses does not guarantee that 100% of the neurons receive only one unique barcode apiece. To address that problem, Church is now working on a way to genetically engineer mice to express random RNA sequences, barcodes in their neurons during development. Though he supports the general direction of the project, Karel Svoboda, a neuroscientist at another research place in Virginia, is skeptical that barcoding strategy or its main competitor, improved electron microscopy, can fulfill IARPA's lofty ambitions. He suspects the mapping will contain too many errors, and Svoboda questions the premise of microns. Given that most important brain functions are distributed across several brain regions, devising useful or fundamental neural computations based on the connection maps of a cubic centimeter of cortex is a pipe dream, in my opinion. If the brain has stereotypical rules that govern cortical circuitry, a cubic millimeter is sufficiently a large chunk of brain tissue that it should give a good picture of how neurons are wired to perform complex computations. We believe and hope that there is this modularity. All of neuroscience is banking on it. If not, microns may be the first to produce solid evidence of that disappointing fact. Yes, because this is what our science does. Yes, this is who they are. Now I know that seemed like a lot of mumbo jumbo, but mRNA, the most recent vaccine, that wasn't the first mRNA. It was in the tetanus. I did a video on it. So by the time they tell you they've already done it on the public, which would then prove the Bible about everybody takes the mark, because that's what we are to them, is a barcode. So those fluorescent lights, maybe they're used for something else, so they know which barcodes can buy and sell.